Welcome to today's IBH seminar. I'm Michael Bayer from the James Hutton Institute. I'll be hosting this for today. So today we have Patrick König from the IPK in Germany talking about PanBARLEX, the web portal for the pan-genome. So some of you might remember Patrick gave a talk last year, about a year ago, on PanBARLEX. But a year is a long time, and Patrick's been very, very busy adding new features to PanBARLEX. So we thought it would be a good idea to get an update from him. And that's exactly what today is going to be. So I'll just introduce Patrick briefly. So Patrick studied hydrology at Dresden University. He then worked as a freelancer for web development until 2017, and then joined Uwe Scholz's group at the IPK in Gartersleben. So he's a research assistant there. He's a research software engineer in the Bioinformatics and Information Technology group. And some of you might know him as the person behind the Bridge portal and also the DiffBrowse web application. And currently he's working on the Shape3 project and also on the Fair Agro project where he's a work package leader. So with that, we should maybe just quickly go over the housekeeping. So we're going to have Patrick's talk, which will be about 25 minutes. Then we'll have a question and answer session at the end. You can put your questions into the chat box at any point, and then we will go through them afterwards. Alternatively, when the talk is over, you can raise your hand. There's a button for that at the bottom. And then we will let you unmute during the Q&A session, and you can ask your question directly. We're recording the event, but the way it is set up now, you don't need to switch off your cameras anymore now. I think that's everything. Malcolm, correct me if I've forgotten something. And if
we're all good.
Yeah, if we're all good, then Patrick, it's over to you.
Yeah, thank you very much for this kind introduction and for inviting me. And okay, let's start. I hope it's working now. Yeah. Okay, what's the motivation behind PanBARLEX? So a short recapitulation why we built up this web portal. And so the barley pan-genome consists of 76 genotypes. And for these 76 genotypes, we have roughly 3.8 million predicted genes. And for those all those genes, we have many sequences, roughly about 19 million sequences, so meaning transcripts, coding sequences, and protein sequences. So it's a lot of data, and the goal was to democratize the access to the barley pan-genome version 2 data via an easy-to-use web portal for all people who are not very skilled skilled deeply in bioinformatics, so wrangling with all the data files, etc. Meaning, I think people from the wet lab, for example, are our target audience, and all people who just want to get a fast insight into the data of the barley pan-genome. Also, the portal should serve as a one-stop shop to check allelic diversity of coding sequences and proteins, presence-absence variation, copy number variation of genes, and also gene expression data. And it provides useful tools for ad hoc analysis and also export features to export gene sequences and multiple sequence alignments of homologous genes. And now the related paper is published since half a year. And so the last seminar I gave 1 year ago, so this was an early prototype of the data portal, of the PanBARLEX portal. And since within this year, I added a lot of new features, and also we discovered some issues with the initial autofinder clustering of the genes. I will come to this later. A little background about the data generation. So we had the 76 genotypes, and all of those were sequenced with PacBio hi-fi sequencing, and 76 chromosome scale assemblies were the result, which end up in 76 FASTA files. And for 20 of the genotypes, RNA-seq and iso-seq data was generated. And this then was used for 20 evidence-based gene predictions done by the PGSP group at Helmholtz Munich. And they used the 20 evidence-based gene predictions as a data resource to project those genes to the 56 remaining genotypes without RNA-seq and iso-seq data. And as a result for this, 76 GFF3 files containing the gene predictions. We get also 76 CSV files with functional annotations and 380 FASTA files with sequences related to the genes. So this is a lot of data, and it's a lot of work to handle this data and to get or to retrieve insights out of this. So this was the motivation for us to build an easy-to-use web portal. And for the 3.8 million genes we have in PanBARLEX, we have gene card pages or detail pages for each gene, where you can see everything important for this gene, the position on the chromosome, the structure regarding intron, extra. You can see function annotations directly here. Also, you can see sequences and export them to your clipboard, or you can download them. And so this is a very basic stuff. This is nothing special, but this is only the basis for everything else. Another problem regarding gene annotations is that we have an evolving gene ID problem. So here as an example, we have 5 gene IDs from 5 different Morex versions or gene annotation versions of Morex. The problem here is that all these 5 gene IDs represent exactly the same coding sequences, in this case of alpha amylase. And this is an issue where we get a lot of requests by biologists, etc., because they are confused about the many different IDs for the same genes, etc. So to overcome this problem a bit, we developed a multi-directional gene ID liftover between 5 different gene annotations for Morex. So on top you can enter a gene ID from these 5 different Morex gene annotation versions, and then you get a result, a mapping of this gene ID to all the other 4 gene annotation versions. And here, this example here is exactly what I showed the slide before. This is alpha amylase, and here you can see the coding sequences across all gene annotations are 100% the same. Another example is shown on the next slide. Yeah, here. And here, for example, you can see I provided the Morex version 3 ID, and a mapping could only found in the pan-genome version 2 annotation. And here you can see that the mapping is also not perfect. So we see here that the target coverage, that means the coding sequence in the Barley pan-genome version 2 seems to be shorter than in the Morex version 3 annotation. So this could be to another length of the coding sequences due to differences in the annotation pipelines, for example. So everything here should be considered with a grain of salt. as all mappings and leftovers, but we think it's also a very useful tool to directly check if an old gene ID is also present in the latest annotation versions. A score for each mapping is calculated by multiplying percent identity, query coverage, and target coverage. So a score below 100% indicates that there's a mismatch between the coding sequences regarding length or percent identity. Also, this multi-direction leftover tool is available in a style where you can enter multiple gene IDs of different annotation versions, and then you can convert all these gene IDs to a specific annotation version. So this is useful for bulk ID conversion. And the leftover was calculated using the easy IBH module of MM62 with a high sensitivity threshold using the coding sequences. And this was done for all pairwise permutations of the 5 Morex gene annotation versions. In the literature, it was shown that MM62 is from the precision roughly the same as BLAST. So it could be used as a replacement to BLAST, but it's very much faster. And this is then handy if you want to calculate many mappings. So yeah. Then another big question was how to then organize the 3.8 million genes into digestible and meaningful groups. So users want to see allelic diversity for the genes across all genotypes, or presence-absence variation, copy number variation for specific genes. And ideally, one can group all the 3.8 million genes into autologs and paralogs, for example, or at least into homologs. And there we first used then AutoFinder to group the genes into autologous groups. This is also known as autogroups. And yeah, so AutoFinder here, the concept of AutoFinder is displayed in this single figure here. It's a very complex algorithm and piece of software. It was mainly developed to analyze phylogenetic ontology across the tree of life. So at least it's in the paper stated. And yeah, it was then applied to the Barley pan-genome version 2 dataset and resulted in 95,000 hierarchical autogroups, whereas 14,736 are single copy hogs, meaning hierarchical autogroups, roughly 2,000 multi-copy hogs, and 80,000 so-called shell hogs or accessory genes, and roughly 500 cloud hogs. When you think about these numbers and you divide the 3.8 million genes for all 76 genotypes through 76 genotypes, you end up with 50,000 genes in mean per each genotype. And so the number of 95,000 autogroups seems to be too high. And even the number of shell hogs or accessory genes seems to be very high. So this was the first bad feeling, maybe. And then we got, yeah, we looked deeper in the data, and we tried analysis with already known genes. And here we tried then VRS1. We blasted VRS1 against all the 3.8 million genes, and then the result showed us that there are 2 autogroups which have good hits with the blast with the coding sequence of VRS1. And so The problem is that this split of a single gene into different autogroups can lead to false positive assumption of presence-absence variation. And here you can see two autogroups which are related to VRS1, and both autogroups are the presence-absence patterns of those both autogroups are inverted to each other. So this is a kind of overclustering, and the same gene is split by allelic patterns into different autogroups, which is a problem because, as mentioned, it leads to false positive assumptions of presence-absence variation if you don't know that these two autogroups are related. And another problem is we normally want to see allelic diversity of the same gene in a combined multiple sequence alignment, for example. And this here is a problem because it's split into two autogroups, and then we have two different multiple sequence alignment of both, and this brings us in trouble. So the reason for the split is that in VRS1 there's a single base pair indel which induces a frame shift, and this ends up that the protein sequence diverges after this single base pair indel diverges a lot, and the applied threshold for sequence similarity in AutoFinder, so there was used the standard settings, could be too hard. So the threshold is too hard, and it divides then the same gene into two different autogroups. And here is the coding sequences, a multiple sequence alignment of the coding sequences of VRS1 and a multiple sequence alignment of the protein sequences of VRS1. And as you can see, the protein sequence diverges more compared to the coding sequence. So in genes with frame shift mutations, coding sequences could be less diverse than the translated protein sequences. And this leads to the hypothesis that coding sequences are probably better suited to cluster the same genes across genotypes in a pan-genome. We saw also with other genes problems, for example, here in a gene family with the function annotation Chalcone synthase, we saw the same coding sequence and protein sequences split into many autogroups. And sometimes there are also autogroups here, the bottom one, where you have the same gene or the same sequence as in the others, but then there's another gene which differs just in one amino acid or in three base pairs. So the whole grouping seems to be too small and to overcluster it. Another problem with the AutoFinder results was that many thousand genes were not assigned to hierarchical autogroups. And so I analyzed this, and in the whole AutoFinder results, roughly 76,000 genes were not assigned to HOGs, meaning hierarchical autogroups. And this could also lead to false positive assumption of presence-absence variation at the end. This problem was also raised up in the GitHub issues here 5 years ago, but yeah, the developer of AutoFinder didn't answer it until now. So the problem is known, but there's no clear answer why this problem happens with AutoFinder. Then I checked the AutoFinder results with many other known genes. And in this table, you can see, so first I checked the presence of the genes in all 3.8 million genes with BLAST and extracted the number of hits in the first column. And then I wrote a script to analyze the AutoFinder results and to see in how many autogroups the gene is present or split. And all the cells here, which are highlighted orange, indicate that a single gene is split into multiple autogroups. And also it indicates in this column number of HOC equal NA. It shows how many of the genes were not assigned to autogroups at all. So this was the problem I showed in the slide before. So, and as you can see here, for just a small set of known genes, we have 1, 2, 3, 4, 5, 6. No, this is the 6 is a Busco marker, which was also split into different autogroups. So, but for these genes, we have a high amount of artificial splits into different autogroups. So, as a solution, we tested a new clustering approach using MMSeq2 software. So MMSeq2 is also used by UniProt to cluster proteins into non-redundant protein clusters. So these are the UniRef protein clusters. And yeah, we did this clustering with MMSeq and end up with 32,000 coding sequence clusters in total. And here I integrated a comparison with the AutoFinder results. So we get a slightly less number of single copy core clusters or genes. We get a higher number of multi-copy core genes or gene clusters. And we get a far way lower number of shell clusters or accessory genes. And also a slightly lower number of cloud cluster genes or unique/private genes. We then also evaluated this with the known genes, and it was then we discovered that the split of the same gene into different clusters/autogroups does not appear with the MMSeq2 clustering. So the parameters for the MMSeq2 clustering were evaluated iteratively. So I started with a very high thresholds, and then evaluated the results of the clustering with analysis scripts, and then it end up with this setting here with a coverage of 20% and a minimal sequence identity of 90%, which gave the best results and which avoided the split of genes into different clusters. So here's the whole pipeline, how I handle the coding sequences or the genes of the barley pan-genome. So first there's a sequence clustering. After this, the sequence clusters, then a multiple sequence alignment is applied with two different softwares, MAFT and Cluster Omega. And the multiple sequence alignments then are used for phylogenetic tree inference with FastTree and Cluster Omega. Also, the clustering and the multiple sequence alignments are then used to calculate cluster metrics. For example, yeah, I will show you the example later. All the coding sequence clusters are available as a browsable and filterable list. And on this page, for example, you can see the cluster ID, then the number of transcripts, genes, and genotypes, the functional annotations of these genes in this cluster, and the physical positions of all the genes on the chromosomes. A cluster metric is, for example, the structural variation index. So this SVI indicates how diverse the coding sequences or protein sequences of a cluster of a coding sequence cluster are. And here are examples. So meaning an structural variation index of 0 means no structural variation at all. And a number near 1 indicates a very gappy and fragmented multiple sequence alignment. And here are some examples. In the first example here, there's one little single base pair indel, and this leads to a number, a structural variation index of 0.01. So yeah, this helps us to see, to directly see how diverse the clustered genes are. Here's a little formula how this separation index is calculated. So basically, it calculates the number of gaps in the columns of the multiple sequence alignment and also the deviation of the sequence length from the mean, and then both numbers are integrated into a metric between 0 and 1. And this structural variation index can be also used to filter the coding sequence clusters. For example, here I set a filter between 0 and 0. That means all coding sequence clusters that have no structural variation, even not a single base pair indel, for example. And here we can see from the 32,000 clusters total, we have roughly 10,000 clusters which have no structural variation at all. But there could be allelic diversity like SNPs, for example. So the structural variation index does only account for indels, but not for SNPs. Also here, I changed the filter to just show single copy core genes with a structural variation index of 0. And here we can see we have 5,262 clusters, single copy clusters, which have no structural variation at all. Also, the interactive multiple sequence alignment viewer was improved in the last month. And it now shows SNPs here indicated with these little stars. And you can switch between coding sequences and protein sequences. And you can also switch between the two softwares used for the multiple sequence alignment, MAFT and Clustal Omega. And also you can here switch to show the multiple sequence alignment only for wild genotypes, for landraces, or for cultivars. And there you can see then the differences regarding SNPs between the wild genotypes and the cultivars. So mainly in the most coding sequence clusters, we see the expected loss of allelic diversity from wild types to cultivars. Yeah, also we have a phylogenetic tree viewer, which is based on the multiple sequence alignments. And yeah, here you can also switch between different options. Then the Barley pan-transcriptome data, which was released or published 3 months ago, was also integrated into PanBARLEX. And it's available on all the coding sequence cluster detail pages. And here you can see an example. And yeah, it's basically just a heat map. And it shows you the TPM values for all replicates and for the 5 tissues. And I remapped all the transcripts to the gene models of the Barley pan-genome. So these gene models are different from the gene models in the PanBARLEX data portal of James Hutton. Also here on this page, there's a visualization of the alignments of all the transcripts. And it's also to only show antisense transcripts. And antisense transcripts can play a role in regulatory of gene expression. And so this could be also useful for scientists or colleagues who are investigating in non-coding RNA. And those antisense transcripts are a class of non-coding RNA. Yeah, here's an example for VRS1, which controls the spike architecture, and yeah, it's a valid result that the gene is mainly expressed only in the inflorescence tissues. Also here we can see for 2 genotypes here in this case, for Barker and for OUN 333 genotype. And yeah, this is also maybe useful for people who are interested in isoforms. Also, the sequences of the transcripts and the TPM values per each tissue and replicate are available as tables. A link to the PanBARLEX portal of James Hutton Institute is also integrated. So you can switch between both portals and the style of mapping to the reference genomes. Data can be downloaded for each coding sequence cluster on the data download tab. So here you can download unaligned sequences as multi-FASTA files, or the aligned sequences, which are multiple sequence alignments with MEFT and cluster omega. On the start page, there's a box to search function annotations of all the 3.8 million genes, and then you get a nice result table with your search word or keyword highlighted, and download the results as a tab-separated value file. And also relatively new, it was added in February this year. We have now a page with known genes, and these known genes are directly linked to the coding sequence clusters. So this is a very handy entry point to study allelic diversity of already known genes or published genes. A feature which is now a little bit older is the interactive pairwise sequence alignment with the Smith-Waterman algorithm, where you can directly in your browser create alignment between two genes, sequences of two genes, and can study with few clicks allelic diversity of two genes. Also, when you are on a gene of Morex, there's a link to the SNP data, which was also published with the Barley pan-genome paper. And so this leads to a diff browse instance where you have access to roughly 150 million SNPs and indels for 1,315 genotypes. But the only link between both data sets is the Morex gene annotation, because the SNP calling in this VCF file was done against the Morex reference. Also, there is a tool to directly export sequences from the chromosome scale assemblies, where you just enter the genotype, the contig or chromosome, and the start and end position, and then you get the according sequence, and you can directly copy it to your clipboard. Also, a tool integrated is a dot plot generator, where you can create self-aligned and pairwise aligned dot plots. And this can help to evaluate copy number variation of genes, for example, or to get an insight what is going around the gene, if there are many repeats, etc. An outlook to future features in PanBARLEX is that I want to use protein language models to visualize the protein space of the Barley pan-genome. And for this, the representative sequences of each of the 32,578 coding sequence clusters are translated to the amino acid sequences and then are embedded with a protein embedding model called ProtBird, which was developed by people at TU Munich. And this ends up with a high-dimensional feature space of the proteins, which can be then embedded into 2 dimensions for interactive visualization. And this is how this looks. So here, each dot in this scatter plot stands for a protein. And here, for example, I have all genes/proteins which are functionally labeled with cytochrome. And as you can see here, all the proteins with cytochrome function annotation, or many of them build a kind of a cluster. some of them are also dispersed in a bigger cluster here. And here's on the left side another example where all genes are highlighted which have the functional annotation pentatricopeptide. And on the right side, all genes are highlighted with which are labeled with resistance. So these are so here you can see that many of the resistant genes build a huge cluster, which which is kind of expected because evolutionarily it's clear that many of the resistant genes have a common ancestor, and therefore they are also very similar regarding their structure. But here, for example, is also another cluster far away from this bigger cluster, which is also a resistance gene cluster. And also there are some genes dispersed in many other clusters. And in future, it would be interesting to evaluate why this is the case. Maybe it's because of misannotation of the function annotation, or it's because of an error in the training of the protein language model. Maybe it could be also the reason. And here, this slide, you can see, or here I overlaid the protein embedding with the expression values, with the TPM values of the transcriptome data set. And here you can see for the different proteins when they are expressed in, or how they are expressed in the different tissues. So sometimes the clusters are, for example, here is a cluster here on top is in bluish color, which means it has low TPM values. And if it's yellow, then it has high TPM values for this tissue. And this visualizes the pattern of expressions on a basis of protein similarity. So in future, the goal is for PanBARLEX to create an interactive tool which can be used to visualize your own ideas or your own functional annotations. And another example here, for example, here's a cluster on the bottom, a small cluster with only a few genes. And these are all the genes related to starch synthesis in the, and this is expressed mainly in the caryopsis tissues. So it, and on all the other tissues, this cluster is not active, or it's not very high expressed, or not expressed at all. So this is a kind of alternative way to study gene expression. And here you can see the whole protein space in one picture. I'm coming to the end, and so the access statistics for PanBARLEX may be also of interest. So we have the United States with the most users currently, and then Germany, United Kingdom, and China. And then there's a drop of visits from all the other countries. The number of visitors and unique visits and unique visitors is still increasing, which motivates us to add new features and also in future maybe new data sets. Okay. I want to thank all the people involved in the Barley pan-genome version 2 consortium and all people involved in the Barley pan-transcriptome project for providing the expression data set. And special thanks to the PGSB group at Helmholtz Munich for a conversation about the AutoFinder results and for providing the gene annotations for the Barley pan-genome. And also special thanks to Martin Mascher, Niels Stein, and Uwe Scholz at IPK for making everything possible. Thanks.
Okay, thank you very much, Patrick. That's been a really good tour around the whole thing. We've got, so far, not very many questions in the chat. Does anyone want to raise their hand, and then we can unmute them and they can ask questions directly? There's a button at the bottom of your WebEx window which says raise, and it's got a hand symbol on it. You can use this. So let's see. So Laura Rossini has got a hand up. I'll see if I can unmute you. Laura, do you want to ask a question directly? It's also
Yes, thank you.
Okay.
Yeah, can you hear me?
Yes, we can hear you.
Hey, hi, thank you so much. This is amazing work and really helping a lot of people to make better use of these resources. So I had a look at the known genes page, and obviously it's just probably a start, what you've got there at the moment. I was wondering,
Yes.
I was wondering how you're planning to enrich it and make it more comprehensive. I mean, are you hoping to get input from individual groups, or you already working on it, or how to move forward with that?
Yes, thank you for this interesting question. So we have an extended list at IPK internally with many other known genes, published known genes. But this is, yeah, the thing is, there are every year there coming new genes, or there are new genes investigated. So I think it's not a problem to send us an email when you discovered a new gene. Send us a mail with the coding sequence or with the link to the paper. The other ideas are that we scan literature in ourselves maybe every 3 months, and then add this in an ongoing process to add the new genes published.
Yeah,
but
and
this
also,
yeah.
That sounds good, but of course, yeah, I think also knowing we can contact you to maybe send suggestions of genes that may not be in your list, or maybe they already are, but just to make sure they're not overlooked. That's great. And the other thing is, sorry, maybe I missed it because while you were presenting, I was also looking at the database. Are you covering also mutant alleles or not?
You mean mutant genotypes?
Yeah, exactly. So for a given gene, you have the diversity obviously in the pan-genome sequences, right? The allelic diversity, but there's also the allelic diversity in mutant stocks. So maybe I didn't catch it, if this is covered or not.
It's not covered by the data portal itself because it focuses on the 76 genotypes of the published in the paper and which are under investigation of the Barley pan-genome consortium. But if you know the gene, you can download all the coding sequences unaligned, merge them with the coding sequences of your mutant stocks, and then recalculate the multiple sequence alignment on your own to see allelic diversity on the coding sequence level.
Yeah, that's very interesting as well to see, the background of the mutant allele. But yeah, okay, so this could be also something in the future to link to some kind of additional information about alleles present in mutant stocks, so that a scientist that's interested in working on that mutant, on that gene, can see all the available diversity, both natural and induced somehow.
Yeah, I think the problem with the mutants is there are many, and it would be introduce many more genotypes into the data portal, and it's then another big story of development and data processing, etc. But as I mentioned, you can download the coding sequences of the coding sequence clusters and then integrate it on your own, which also saves you maybe a lot of time.
Thank you.
So just to follow up on that, so is there mutant data in DiffBrowse that you can link through to from PanBARLEX? Because you're linking to DiffBrowse already.
Yeah. So the link is via the Morax genes. but I don't know, in the diffbrowse instance, we have 1,315 genotypes, but I don't know if there are mutants in this data set. So I'm sorry, but you can, yeah, directly see in the diffbrowse instance which genotypes are there. But I know that 1,000 of the genotypes is the core 1,000 set of the Bridge project. So these are gene bank material, and these 315 other genotypes are cultivars from breeders. Yeah, but I don't know if there are mutants within these 315 cultivars of the breeders. I don't know.
Okay. Thank you, Laura, for the questions. So no one else got a hand up at the moment, so there's questions in the chat here. Lots of thank you messages, first of all, Patrick. I don't know if you can see the chat. Then,
yeah, I see the chat, yeah.
Okay, so
Thank
then
you.
there's a question from an unnamed attendee saying you have generated a lot of sequences from the 76 cultivars. From a breeding perspective, how do you think your data can be translated into primary and useful information tools for serial improvement?
So that's a good question and not easy to answer. So the main goal of the data portal of PanBARLEX is to provide easy access to the pan-genome data. And I think the idea is how to translate the data and the allelic diversity, etc., to improvement or into breeding improvement is more on the side of the breeders. So yeah, currently, I think maybe in future we can add maybe tools. So we have a follow-up project at IPK of the SHAPE project called Pre-Breed, and in the next 3 years, and there's maybe the possibility to add special tools for that into PanBARLEX. But currently, it provides the data and tools to study the diversity, and then in the hands of the user to extract the relevant information and to transfer it into breeding, I think.
Okay. Yeah, still no one else got a hand up. So we'll just take the next question from the chat. So Andrea Pacello asks, thanks for the presentation. Are there tables associating Morex gene IDs from V3 or V2 genome versions with the respective PanBARLEX Morex gene codes available to download in case we would like to lift over large lists of gene IDs?
Yeah, currently we don't have a bulk download for this list, but yeah, we can add this. So maybe look in 2 or 3 weeks, and then we put it in the download section. Yeah.
I think you also had a function to paste a list of gene IDs, didn't you?
Yeah,
And
yeah,
get them translated.
we have a function to paste a list of gene IDs to have a bulk conversion. But I don't know if you enter 10,000 gene IDs, it maybe does not scale to this. I tested it with 1,000 gene IDs, but yeah, I think to incorporate everything into the pipelines, into your own pipeline, it's better to have a mapping or a file, a table, a comma-separated value file or similar. But we can add these files to the download sections. Yeah.
Okay. So the next question is saying, I have a question concerning the PanBARLEX site. When I try to find a phytase gene using the ensemble plant gene ID, I can't find any gene. Why? I presume that's the ensemble plant Morex V3 gene ID.
Yeah, I think maybe the ensemble plant gene ID is not in our database, or it's not it does not fit to the gene IDs or the gene lift over tool. So in this case, I recommend to extract the coding sequence from ensemble plant and then directly using the blast feature of PanBARLEX to get to the related genes.
Yeah. Okay, then here is a comment from Robbie saying, Robbie, do you want to unmute and talk through it?
Hold on, give me a second. I'll unmute Robbie.
Okay.
Sorry, should I answer Robbie's question, or is it more a discussion? I don't.
Well, no, I think we're going to unmute Robbie, but maybe he's not. Maybe there's got issues with the microphone. So I'll just read it out. So Robbie says it would be great to be able to search with known gene names. And you gave the nice example of VRS1, which in most databases, if you search for VRS1, it returns no hits. And there are, I'm sure, lots of others like this. Would there be any community interest in federating the annotation of gene sequences in the database through, for example, an annotation consortium? So, yeah, I mean, that's a question for all of us, really, isn't it?
Definitely, yeah.
I think we're all in the same boat with this. We're all struggling with it, and yeah, we're all looking for solutions for that. I think this is a big topic, and we may need to address that separately at some point.
Yeah, I think this goes also in the direction of the gene ID problem. And I think this is one of the million-dollar questions, how to get stable gene IDs across different gene annotations reliable. So yeah, we all know in Arabidopsis, they tried it, but at the end it was not successful at all. But this is the same problem is with the functional annotations. So in my opinion, there must be a curated database with a community effort, which is curated collaboratively by all Barley people. Yeah, this would be, I think, the most appropriate way, because then you can spread all the workload across many people. Yeah.
Okay, so I don't see any more questions in the chat. Let me just check the participants list, see if anyone's got their hand up. No, don't see anyone there. There's one more comment in the chat from Laura. Yeah, there's a bit of discussion.
Yeah, that's the problem with they're discussing about the gene names. So on the known gene pages, this page is curated independent from the functional annotations. So when you search the functional annotations, it searches all functional annotations provided by the annotation of PGSB group, Klaus Meyer, Manuel Spannagel. And there, sometimes you don't find the gene by its name because it's simply not in the databases where they retrieve the functional annotations. So if VRS1, for example, is not annotated in UniProt, you cannot find it via this way. And that was also the reason why we created this known gene page. But this is then hand curated, yeah, and which is also then a lot of effort to curate and yeah, this table or this data set.
Yeah,
it's not impossible, but it has to be done, and it takes time. And I think it would be in future nice to have a collaborative platform where all Barley people can contribute to the annotations, etc.
Yeah, there are discussions ongoing at the moment about having something similar to what they have for Arabidopsis, where you have community curation, you can have gene names, and you can have multiple identifiers, and you have potential input from members of the community. So I think something like that is definitely needed going forward, because this problem is only going to get worse, isn't it?
Yeah, definitely.
Okay, right. So I'll have one last check if there's any more hands up.
Yep, Uwe Schultz put his hand up, Misha. If you can unmute.
Yeah. Uwe, it's all yours. You can speak. We can't hear you. Uwe,
Here we go.
Uwe, can you hear us? We can't hear you if you're asking a question.
His mic's on.
Yeah.
Hear me now?
Yes, now we can hear you.
Okay, then I had some problems with my microphone. I will add a comment regarding the discussion about finding annotation and so on. And there was this community effort discussed and so on. But I think we need really in the Barley community a group which is financed by in-house money from the institutes which are contributing, and otherwise we have no sustainable solution for such things. And from my point of view, the institutes like James Hutton, IPK, and so sit together and look what is possible to set up such a annotation group. I think the community effort, we tried this in the old PanBARLEX system, and this was implemented by a guy, his name is Burkhard Steuernagel, that the people can add annotations, but nobody used it, I think 10 years or 15 years ago. I think there should be a really responsible persons who checking this, look on it, make quality checks and curate it. Otherwise, we cannot come to a sustainable solution. This is my opinion, and sorry for put this here into the round.
No, thank you. That's a good kind of suggestion, and I think we will have discussions about this certainly within the framework of the people on the call here in the near future. So yeah, thank you for that point, Uwe. I have a question, Patrick.
Yeah.
Perhaps the last one. So what I wasn't clear on was is everything you have in the portal now based on your new clusters that you have generated with MMCX2, or is some of it duplicated where it looked like in your multiple sequence alignment viewer you could choose between the clusters generated with AuthorFinder and those generated with MMCX2, is that right?
Sorry, I didn't get your point.
So at the moment in your PanBARLEX interface,
Yeah.
is all the information that you have now based on the new clusters that you have generated with MMCX2, or is there duplication between that and the old clusters that were generated with AuthorFinder?
Yeah, so currently both strategies live side by side. So
Right.
the
Okay.
auto group clustering is still available, and it's independently from the MMCX2 clustering.
Okay.
But I started to implement new features only for the MMCX2 based clustering, because, for example, to calculate metrics, etc., I need to go through all the clusters, which takes a lot of time, and I see no reason currently to push the AutoFinder results forward. And we discussed to remove them because they could lead really to confusion. They are still there, but I think we will remove them or in the upcoming weeks. So because it was definitely a try, but it turns out that the results were not very reliable, and
Okay.
Best option is currently to use really the coding sequence clusters by MMCX2.
Yeah. Okay. Well, I think that probably brings us to the end now. One last check. No one's got their hand up. So I think if there's nothing else in the chat either, then we'll draw this to a close. I would like to thank Patrick very much for an interesting talk and a great tour of this great resource. And if you have missed some of the presentation, it will be on our IBH YouTube channel, the whole thing, the recording. And I think that's pretty much it. We can thank Patrick by using the clapping thing that you can get at the reactions button at the bottom if you haven't seen that before. So thank you very much, Patrick.
Yeah, thank you for having me. Thank you very much.
You're very welcome, and we'll hopefully see everyone soon at the next IBH seminar. Thank you.
Goodbye. Thank you.
Bye-bye now.
Thanks, everyone. Thanks. Bye.
Thank you. See you later.